Prokaryotic cells come in different shapes or forms. We're going to make a list of the 3 most common forms. And a lot of times the shape of the bacterial cell, that shape is reflected in the naming of the bacteria. So, for example, we were mentioning Staphylococcus Aureus. Staphylococcus is an example of a coccus-shaped bacteria. Coccus means spherical. So our first one to put down on the list is coccus or cocci. Cocci is plural. The cocci are spherical in shape. So the cells just look like little spheres. Okay. Other bacteria, instead of being spherical, they're sort of elongated, and they look sort of like little rods. The bacilli. Bacillus is the name for this type of bacteria. Bacillus, or plural, is bacilli. The bacilli are rod-shaped, kind of like so. So instead of being spheres, the cells are elongated. And then the third type that is common amongst bacteria are the spiral-shaped, or spirillum. Spiral-shaped bacteria, they are spiral-shaped. Spiral-shaped, and so these are kind of interesting. These look like sort of, they have a corkscrew sort of a shape, so spiral-shaped. It's kind of hard to draw on a flat surface, but anyway, spiral-shaped. Some of these are disease-causing. This is a good example of this is the bacteria that causes Lyme disease. It is a spiral-shaped bacteria. So 3 different common shapes for bacteria. We're going to make a few headings here. Still talking about prokaryotic cells. How do prokaryotic cells reproduce? So we spent a chapter talking about cell division, and we were focused in on eukaryotic cells primarily. In the past, back when we were in person, talking about cell division, we talked about mitosis and the different stages of mitosis. That deals with dividing up the contents of the nucleus, which is an organelle. In prokaryotic cells, there are no organelles to deal with, so cell division is a little bit different. Cell division happens by a process called binary fission, and this is generally how prokaryotic cells reproduce. So we're going to say reproduce by a process known as binary fission. And binary fission is really quick. It doesn't take very much time. In this process, what happens is it's kind of analogous to when we're talking about eukaryotic cells. Basically, the DNA needs to be copied. So the cells copy their DNA. And then they divide their contents, the contents of the cell, in 2. Copy the DNA, then divide. And again, this is very quick. This doesn't have to take very much time. Oftentimes with bacteria, they can divide in just like 20 minutes, some only 20 minutes. There are a few bacteria who can go even quicker. I think the shortest amount of time that I recall off the top of my head is like 5 minutes has been measured for the amount of time it takes for bacterial cells to divide. So you can imagine bacterial populations, they can grow large very quickly if they're able to divide that fast. As long as they have resources available, as long as they have a good food source, the nutrients that they need, they can multiply very quickly. So in the process of copying their DNA, this is done by a polymerase, just like in our cells, but it's not uncommon to have mistakes along the way. In prokaryotic cells, when they copy their DNA, spontaneous mutations can occur. And so that leads to, you can probably guess what, leads to a lot of genetic variation. There's a lot of diversity amongst microorganisms. Lots of genetic variation. Okay, and then sometimes that genetic variation can lead to different things, such as antibiotic resistance. So bacterial populations in the context of human health, if a person has an infection with a bacterium, then antibiotics would be given to treat that infection. But if the bacteria happens to have mutated, then it's possible that the bacteria will no longer respond to the antibiotic in the expected way. And that's something that can arise through these genetic mutations that take place. Antibiotic resistance is a good example of a consequence of this. Okay, so binary fission, again, it can happen quickly. So this actually brings us back to the topic of refrigerators. Why do we have refrigerators? Cooling our food down, all this is doing is slowing down the process of binary fission. So refrigeration, it does not kill bacteria at all. Rather, what it does is it just slows them down. So the food lasts longer. It doesn't go bad quite so quickly because the bacteria are behaving a little bit more slowly. So refrigerating food specifically. Refrigerating food. What this does is it slows the rate of binary fission. And so note, it does not kill the bacteria. Okay, all right, so let's talk about nutrition for prokaryotic cells. What do they need in order to survive? Nutrition. Okay, so nutrition. Some bacterial cells are capable of photosynthesizing. So some are photosynthetic, similar to plants. Photosynthesis is possible for some. Others rely on organic matter. So it's kind of like they have to eat. They have to take up organic molecules and use those as a food source. Organic matter. Others are capable of living off of inorganic matter, meaning not the biological molecules, but other sorts of molecules. We'll put some examples down in just a second. Inorganic matter. All of these are possible. These are possible nutrient sources for prokaryotes. Okay, so quite diverse. Let's just make a note here. Let me get a different color. And let's make a note about this one. This one's probably not quite so familiar. Inorganic matter. Some examples of this. A good example would be for bacteria that live in the soil, so soil bacteria. They're not exposed to the normal atmosphere like we are, or cells that live on the surface of our bodies. Instead, what they rely on is ammonia in the soil. So an example of an inorganic matter would be NH3. This is ammonia. NH3, ammonia. And this is what soil bacteria rely on. So soil bacteria don't depend on light. They're underneath, they're in the ground, they're below the surface. Soil bacteria, they don't depend on light. Instead, they depend on ammonia in the soil as a nutrient source. So what the soil bacteria do in large part is they support plant life. They help plants to be healthy by maintaining the soil conditions, and so that's a good thing. Another example, let's call this example number 1, and then example number 2. Another good example of inorganic material that prokaryotes can use is hydrogen sulfide, H2s. This is hydrogen sulfide. Hydrogen sulfide. This is something that exists near hydrothermal vents at the bottom of the ocean. And again, there are prokaryotes that live at the hydrothermal vents, a mile down under the sea, and they support animal communities in those habitats. So hydrogen sulfide, this would be an inorganic material that prokaryotes use near hydrothermal vents. Hydrothermal vents. Okay, and while we're on this note here talking about nutrition, there are some bacteria that live in very close associations with other living things. This is called symbiosis. So let's just we can squeeze this in down here. Symbiosis, oops. Symbiosis. This is a word that means living together. Living together. Bacteria often live together with other things. This is something that exists between two or more species, so it's involving not just the same species, but multiple species. Symbiosis occurs between two or more species, and both of those species can benefit from this interaction. Both may benefit. Okay, example regarding bacteria. If you've ever noticed, we've probably noticed outside, sometimes you'll see a rock that has like a colorful thing growing on it. What is that? It's called a lichen. And what a lichen is, lichens are, it's a symbiotic relationship between a bacteria that can photosynthesize and a fungus. So bacteria and fungus. Lichens are consist of, they're called cyanobacteria. They're a type of photosynthetic bacteria. Cyanobacteria, which are photosynthetic. So what they do in this relationship is they photosynthesize. They use the energy from the sun. The bacteria photosynthesize, and what that does is provides food for the fungus cell. The fungus cannot photosynthesize, so instead the bacteria provides some energy for it. So providing food for the fungal partner. Okay. There are many other examples of symbiosis, but that's just one to give an idea, something that's hopefully familiar, seeing lichens on rocks before. All right, one more section here. Let's talk about the ecological impact of prokaryotic cells, of bacteria particularly. Let me go down here, get some more space. Okay, ecological impact. This is our last major heading for this video anyway. Ecological impact. All right, so prokaryotes come in many different varieties, so they do a lot of different things. Prokaryotes, we've already mentioned that there are some prokaryotes that live in the soil and they metabolize ammonia, and this is very good for plants. This helps to make nitrogen available for plants. So ecological impact. Okay, first thing is we're just going to list that specifically. Prokaryotes can metabolize nitrogen in the soil. And what that does is makes the nitrogen available for plants. So the bacteria use ammonia, which has nitrogen in it, break it down, that makes the nitrogen available for plants. And then once the plants take it up, animals can use the plants. So when we when we get into ecology, we'll be thinking a lot about these interconnections between living things. It all comes down to energy flow. So in this case, energy is flowing from the bacteria to the plant, and then from the plant it could flow to an animal if an animal comes along and eats the plant. Okay, another thing that prokaryotes do, and this is a very important job, prokaryotes decompose organic matter. So they're kind of like the recyclers, along with fungi. They recycle things, decompose organic matter. And what do we mean by organic matter? So this could include wastes, literally waste from animals, wastes, and also this includes perhaps organisms that have died. So the carcass, what happens to the carcass? It's a good thing. Carcasses don't just hang around forever. Bacteria decompose them, and then those molecules and atoms can be reused to build other things. So wastes and dead organisms, plants or animals. Okay. And in the end, what this does, after organic matter is decomposed, what this does is it returns elements back to the environment for use by other organisms then. So it allows recycling. And then that kind of leads to another subtopic here. It is possible to use prokaryotes to do messy jobs for us. And this is one thing that is very, very handy in our society. Bioremediation. Bioremediation, this refers to any process that uses organisms, living organisms like bacteria, in order to remove pollutants from a given situation. So bioremediation, using organisms like bacteria to remove pollutants. We'll put a few examples down of this. Okay, examples. A great example of this, bioremediation, would be using prokaryotes to clean sewage water. So sewage treatment, how does that happen? How do we clean dirty water? Sewage treatment plants carry out bioremediation. Sewage treatment. And in the end, what we get from this process is quote-unquote clean water. Okay, so it is technically clean water. This doesn't generally go back into the drinking supply, considering where it came from, but it is certainly clean compared to what it used to be. So clean water. Sometimes, I don't know if you've ever gone to, like, Legoland is what's coming to my mind. I was there earlier in the year. I think I told you all about that a little bit. But anyway, if you ever go to larger cities, sometimes you'll see signs out, like in parks or in theme parks, and the signs will be saying don't drink the water that's coming from the sprinklers. Like, they're using it to water the grass, and it works totally fine for that purpose. It is clean water, clean enough to do that, but they have signs out not to drink it because it is reclaimed water from sewage treatment plants. So kind of an interesting application there. Sewage treatment. Another example would be just toxic chemical cleanup. If there's a chemical spill or if there's an oil spill, how to clean it up after the fact. A lot of times this makes use of bioremediation. So toxic chemical cleanup, using the fact that prokaryotes can metabolize things. That's one way to break down the toxic chemicals. And then again, oil cleanup would be another good example. Okay, so that wraps up the prokaryote section of this chapter. Last thing we're going to do in the next video is go over eukaryotic microorganisms.